Hello everyone, I hope you all are doing well. So for the past few days I have been getting a lot of messages from your side to discuss about my revision strategy for IIT JAM Biotechnology exam, specifically for the last few months. So in this video I am going to discuss few things about revision because it is very important, as it quite naturally the things you have read in your first or second semester or even third and fourth semester, you are going to forget many things you read during that particular time. So it is very important to have a solid revision strategy such that you can properly memorize the things and you can be confident before entering the examination hall. So let's start the video. Now in IIT JAM Biotechnology question paper, there is basically questions from 4 major subjects, that is biology, chemistry, mathematics and physics. And among that, biology, the question paper consists of a total of 60 the question paper consists of a total of 60 questions, among which almost 40 questions are asked from biology. So you can consider almost like 60 to 65% questions are asked from biology, and the rest part are asked from chemistry, mathematics and physics. And among that, chemistry portion is generally maximum, but you can consider chemistry, mathematics and physics will be almost equal in proportion. Now there are 3 kind of question: multiple choice question, multiple select question and numerical answer type question. Now for MCQ and MSQ you have to choose the correct option, and for NAT, these are mathematical question and they will provide you a rough sheet. You have to solve that question in the rough sheet, and you have to type the correct answer with the help of the keyboard. The answer might be a whole number or a decimal number, but you have to type the right answer. Now for the MCQ question there will be only one correct answer, but for MSQ question there will be either one or more than one correct answer, but you have to choose all the correct options. If 3 options are correct but you are choosing only 2 of the 3 correct options, then you will not also get any marks. Or 2 options are correct but you are choosing 3 options, then you will also not get any mark. And for the MCQ question there will be negative marking, but for MSQ and NAT there will be no negative marking. Now let's look at the number division. Now there are basically 3 sections: section A, section B and section C. Section A is MCQ section. There is a total of 30 MCQ, among which 10 MCQs are 1 marks question and 20 MCQs are 2 marks question. And section B is the MSQ section, and each of the MSQ consists of 2 marks. And section C is the numerical answer type question, and there is a total of 20 question, and among which 10 NAT questions are of 1 marks and another 10 NAT questions are of 2 marks. So the MCQ questions consist of a total of 50 marks, and NAT and MSQ questions together consist of another 50 marks. So total of 100 marks. Now for NAT and MSQ, as I have mentioned, there is no negative marking, but for MCQ questions there will be negative marking. So for the 1 marks MCQ there will be 1/3 negative marking, and for the 2 marks MCQ there will be 2/3 negative marking. So for the 2 marks questions, if you don't know the answer, if you are not sure, try to avoid answering that particular question. You can leave that question, because negative marking is very dangerous. Your rank will be drastically decreased. So now let's discuss about the major topics which you should focus on. Now for biology, these are the modules which you should mainly focus on, because these are very important: biochemistry, molecular biology, cell biology, microbiology, immunology, genetics, physiology and endocrinology. So in biochemistry you have to focus on the enzymes, specifically the Michaelis-Menten kinetics, about the enzyme inhibition, about the rate of reaction, that questions. In metabolism you have to focus on the glycolysis, Krebs cycle, about transition reaction, about the oxidative phosphorylation, about fatty acid beta oxidation, about protein metabolism, transamination, deamination. These things are very important. Now in molecular biology, you have to read the central dogma portion, that is DNA replication, transcription and translation, and operon concept, post transcriptional modification, post translational modification, phosphorylation, all that things. In cell biology, you have to study about the cell cycle, cell organelle, about apoptosis, about cancer. These things are important. In microbiology, you have to study about the classification of microbes, about bacteriology, virology, about the growth curves, bacterial growth curves and viral growth curves, about the microbial genetics, about like conjugation, transduction, generalized and specialized transduction, all things. In immunology, you have to study about the innate and adaptive immune responses, and in adaptive immune responses you have to also study about the cellular and humoral immunity, all mechanisms. And in genetics, you have to study about the Mendelian genetics, exceptions of Mendelian genetics, pedigree analysis, gene interaction, like epistasis, dominant recessive epistasis, about the complementary supplementary gene, pleiotropy. You have to study about the mapping the distance between two genes with crossing over frequency, population genetics. These are important. In physiology, you have to study both animal and plant physiology, and endocrinology portion is also there. So these are the major things, and few things are also left, but after completing these major topics, you have to focus on other topics, because these portions has a very high weightage in the IIT JAM Biotechnology exam. Now let's look at the revision strategy. So whenever you are revising, you should first focus on the concept, and then after your concepts are clear, you should focus on the facts and the pathways. And whenever you are reading a particular topic, you should have a basic outline of that particular topic in your mind. The outline should be clear, and whenever you are reading topics, try to relate them, because biology is a single unit and every topic is in any way related to one another. And whenever you are reading a particular topic, whenever you are reading cell biology, try to relate it with molecular biology. I am discussing this topic in detail in the next slide, but till now this relating different topics is very much important, because it will make your concepts clear and will give you another level of confidence, and this is important. Now during revision, you can follow flowchart and diagram from the standard books, but sometimes the flowchart or diagrams are a bit complex in certain books. Therefore, you can make your own flowchart or diagram or certain mnemonics, such that you can memorize things easier. Now let's assume that you are reading protein trafficking. So whatever you have to read basically is like proteins are synthesized in ribosomes. Ribosomes might be present in the cytosol, cytoplasm, or they are bound to endoplasmic reticulum. Now after synthesis, the protein will be transported in vesicle to Golgi complex, and in Golgi complex certain modifications will be there. There will be certain glycosylation, and after glycosylation certain tags will be attached to the protein, and with the help of that some will be directed to lysosome, some will be directed to membrane or other locations. So these are the basic outline. This should be clear in your mind before like going into the detail. Now after the basic outline is clear, you should go into the detail, like what events occur inside endoplasmic reticulum, what occurs inside Golgi complex. Like in endoplasmic reticulum, there are certain enzymes which function in the function on the newly synthesized peptide. There are protein disulfide isomerase which help in the formation of a disulfide bond. There are oligosaccharide transferase which transfers the oligosaccharide, and there is a certain residue aspargine X serine threonine. On the aspargine residue, the sugar will be attached. You have to study about the signal peptide, signal hypothesis, about the COP1, COP2 vesicle, which help in the anterograde and retrograde translocation between Golgi complex and endoplasmic reticulum. So these topics are very important, but you will focus on that topic whenever the basic outline is clear in your mind. I hope you are getting what I want to say actually. Now, in Golgi complex, you can focus on the structural aspect, about the cisternal maturation model, about the vesicular transport model, all that things, about how mannose 6-phosphate residue is attached, how they are directed to lysosome, the structure of lysosome. Now, after that, you whenever this is done, you should focus on how you can relate things. Now, you are reading about ribosomes. So, where ribosomes are synthesized? They are synthesized inside the nucleus. Where? In the nucleolus. So, this is the location where ribosome assembly occurs. So, after ribosome assembly, they are transported outside the nucleus, and nuclear pore complex helps in the transport of ribosome. So, you have to study about the structure of nuclear pore complex. So, these are the topics which you can relate. Why you are reading a particular topic, you should know that. Now, after that, like during protein synthesis, and the information of the protein synthesis is encoded in the mRNA, and based on that sequence, the amino acids will be coming and protein will be synthesized. Now, this mRNA is synthesized inside the nucleus from the DNA. So, transcription will occur, and after that there will be post transcriptional modification. So, after post transcriptional modifications, the mRNA will also come out from the nucleus, from the nuclear pore complex. So, you also have to read about the mechanism of nuclear pore complex, how importin and exportin molecule help in the transport of different molecules between nucleus and cytoplasm. So, these things are very important to relate different topics. So, I hope these things are clear. Now, let us discuss about the source from where you have to read. Now, let us assume that you are such a student for whom the basic outline about the key concepts are clear, but you have just forgot the main informations. Some informations you forgot because it was a long time, almost 1 or 2 years, whenever you were in your first semester or you have read that particular topics. So, if you prefer watching things from videos, so you can refer to lectures, different detailed lectures. 1 second. So, if you are enrolled in certain batches in any platform like IFAS or Physicswala or Bansal Biology or Ved Prabhav, in any platform, you can refer to their lectures. But while watching their lectures, if you are going to make a note of all the lectures, then you are going to take almost 15 days during completing a single module, and that will be a bad decision. That will not be a wise decision. So, I will suggest you during revising that topics from detailed lectures, you just revise things from the PDFs. You watch the lecture, but whenever you are again watching the same thing or again wish to read the same thing, read from the PDFs of that online lectures, because you don't have time to make notes of all that things. For physics, chemistry and mathematics, it is okay to make all the notes, but for biology, I will not suggest you to make all the detailed note up just before 6 months or 5 months before the exam, because that will be going to take a lot of time. Now, if you prefer that you don't like to revise things from the online lecture, but you like some book to revise things, then you can follow Pathfinder Life Science volume 1 and 2. These are very detailed books, but this book mainly focuses on information rather than understanding. The standard books generally focus on understanding rather than information. There are also a lot of information in the standard books, but these Pathfinder books are written in such a way that they collect information from different standard books. There are lots of proteins, name of different molecules, different proteins. Therefore, if your concept is clear during reading a particular topic, then it's okay. And if you are feeling that there is lots of information in the book, then you can skip that information. You just you just read the main things from that book, and you can highlight the book with different colors, red, blue, green, such that the books looks more interesting, or you can memorize things easily, then that will be fine. Or if you feel that your concepts is not clear or your basic understanding is not clear, then you can first clear your concept with the help of one shot lectures. For me, I personally followed lectures of Mamta ma'am during my revision. So here you can see, here this is the one shot revision series of molecular biology for GAT-B exam. But for any exam, the videos lectures are almost same. So you can follow lectures of any exam. The pattern is almost same. You can follow Mamta ma'am or any other ma'am or sir that suits you. So watching the one shot online lectures, you are clearing your concept. Now after your concept, your basic outline is clear, you can move on to either to the Pathfinder Life Science book, or if you prefer reading from like online lecture, you can move on to the detailed lectures. Now during whenever you are like reading the one shot lectures, you are going to face a lots of difficulty, because certain things the teachers will be skipping, or they will not be going to cover the whole things. But for clearing the initial concept, either you can start from the one shot lecture, or you can directly start from the detailed lecture and revise again from the PDF notes. And certain things you can write in your notebook. For that, you can watch the very first video of this channel, the first video regarding IIT JAM, and that video will help you. Now I hope these things are clear. Now whenever your theory portion is complete, you need to practice a lot of questions. Now I personally for practicing questions, I practiced the book IFAS by IFAS publication, IIT JAM Biotechnology previous year questions. So you can complete the book, and almost you should complete previous 15 to 20 year questions. And for this particular book, many questions are given wrong. So if you are feeling that this question is wrong, you can use ChatGPT or Gemini or GorkAI or any other AI tool to recheck the answer. And apart from the IFAS publication book, you can follow any other book, but make sure that the questions are given in chapter wise manner and topic wise manner, and then this will help you. Like if suppose you have completed only one module, and that particular questions will be sorted in the book, and you will be practicing that particular questions only. So this will help you. Now after practicing, suppose you have completed all the previous year questions for IIT JAM, and also for multiple times, for once you are completed, again practice the same questions, and whenever you are done, you can also practice questions of CUET or GAT-B exam. That will also going to help you in IIT JAM exam. Now whenever your question practice is done, like during practicing questions, try to maintain a good notebook for PYQ solutions. Suppose you are practicing previous year questions of biochemistry, and you are solving questions of amino acid. So you have to maintain the like proper question number, and the proper answer should be written in your notebook. And whenever you are facing difficulty for a particular question, you have to highlight that question, because whenever you are going to solve the similar question, you should not fail, you should not like feel difficulty second time, because during the first time you are solving the questions, you are already giving lot of time for that particular question. So if you maintain a good notebook and highlight the difficult questions, it will definitely help you in the long run. Whenever the exam will be closed, only 1 month will be there before the exam, or 15 days will be there, and you just like giving too much effort to complete as much as you can in the short period of time, and on that particular time this notebook will definitely going to help you. So I hope I have mentioned all the points Now if you have any particular question how to complete topics, from where to read certain topics, you can definitely ask me in the comment section or in the Telegram group or in Instagram. I will try to reply as soon as possible. I generally try to reply all the comments. So I hope I have completed the major things, and thank you everyone for your support, for your patience. I hope this video will be helpful. Thank you very much.